Today we are going to start transduction, and transduction is the same topic of like transformation, but in between one vector is there, and vector we know it is the method how the genes or DNA is transferring from one organism to another organism. So the DNA and genes is transferring from one organism to another organism through viral vector. So in transduction, the viral vector like bacteriophage is required to transfer the gene or genes. Okay, so if you see the definition, the bacterium where the one gene is transferring from one bacteria to another bacteria through the bacteriophage, we are calling the transduction. So in simple word, if you will see the gene genetic material, it may be a single gene or it may be a multiple gene. It will be transferred from one organism to another organism by bacteriophage. And bacteriophage is a viral particle which can able to infect the bacteria. So specifically they are infecting the bacteria. That's why we are calling bacteriophages. So many type of bacteriophages is there which will infect the particular type of bacteria, and they are having their own host, different bacteriophage. So they will infect and they will do this transduction. Okay, so when they are transferring their genetic material from host, a host can be a bacteria. So when they will transfer their bacteriophage genes, so bacteriophage genes when they will reproduce and they will able to produce more and more bacteriophage. So in this process they will take up some host genes also, and when they will pack their DNA, entire bacteriophage DNA. So with the host DNA and bacteriophage DNA, their DNA also will be transferred. So with the help of their transferring mechanism, the bacteria DNA or genes also will be transformed from one organism to another organism. So in this process, the foreign DNA means bacteriophage DNA, which is introduced by the bacteria inside the bacteriophage, and then after some time when it will replicate, the bacteriophage DNA genome will replicate. It will take up the small amount of number of gene, one gene or two gene, and then it will transform from one bacteria to another bacteria. So combinedly we are calling transduction this process, and we'll see how it is different. So if you will see Lederberg and Zinder in 1952, they discovered this transduction mechanism in first time in the history. And then there is two type of process. First one is the lysogeny and second one lytic phase. So we are dividing transduction in two type of, and first type lysogeny phase, if you will see the host cell and the bacteriophage, they are having a stable relationship, means host cell will not be lysed by the bacteriophage. Lysis means breakdown of the cell wall and plasma membrane, so that many bacteriophage will be released in the environment. Okay, so this process will not be there, and the bacteriophage DNA will be attached to the bacteria, and it will be replicated again, and the bacteria also can be do fission, so that the number of bacteria will be more, and inside the bacteria your bacteriophage DNA will be there. It may be attached or it may be not be attached, it depends on the condition. Then when suitable condition will be there, so this bacteriophage can able to do can able to turn into the lytic phase. And in the lytic phase, if you will see, the bacteriophage infection will be there, and after the infection bacteriophage will be multiplying, and this multiplied number of bacteriophage can able to do lysis of the plasma membrane and cell wall of the bacteria, so that many phage will be out in the environment and they are ready to infect other microorganisms. So here these two phases is there, and different bacteriophage, they are infecting different bacteria using these two cycles. Now these two cycles, if you will see how it is happening in detail. first we'll see the lytic phase. So in the lytic phase, the plasma membrane and cell wall will be lysis. That's why we are calling lytic phase. So when the bacteriophage is attached to the bacterium plasma membrane or cell wall, it will inject its DNA, and this DNA will be a circular DNA when it is injecting, and after the injection this DNA will convert into the double stranded circular DNA. Okay, because it is having cohesive end. We'll discuss what is cohesive end. And when it will convert into the double stranded circular DNA, it will replicate its own. Okay, when it will replicate, and it will make their multiple copies, and it will synthesize structural component of the bacteriophage, and it will create new phages in the cells, and this new phages will released by the cell lysis. So the bacterium which is having infection of phage DNA, it will die because of it ruptured the cell wall and plasma membrane. So all the content of the bacteria will come out, and new phages will be released in the environment so that they will infect new cells. So this cycle where the cell wall and plasma membrane is lysic, lysis is happening. So we are calling lytic phase, where the bacterium will be died and the new phage will be synthesized. So lytic phase. Then lysogenic cycle is there. So after the infection, when the double-stranded circular DNA will form, this double-stranded circular DNA, it will not uh, come into the lytic phase. What it will do, it will attach to the main genome of the bacteria, and it will multiply with the bacterial genome. When it will multiply again, so population or will increase in the bacteria itself. So number of bacteria cell which is harboring the bacteriophage DNA, it will increase. Okay, so when the bacteria will divide, they will also their genome also will divide, and so many bacteria which is having bacteriophage genome, they will in number will be increased. So in some occasion what will happen, this bacteriophage also release from the bacteria, and they will go inside the lytic phase, and then time they will do the lysis of the bacterial cell. So lysogenic cycle can able to persist from the some time, and then they can able to go for the lytic cycle. Okay, so when they are when they are making these lytic phase and lysogenic phase, so when they are synthesizing new bacteriophage, these bacteriophage can able to carry the small portion of the genes, some genes they can take inside the bacteriophage, and when they will transfer, and they will inject or they will infect to the new bacteria. So what will happen? The another bacterial genes will be transferred into the infected bacteria, and then what will happen? The genes will be integrated into the bacterial gene. So here, what is the important things? The in the infection is okay, but the bacteriophage DNA which is carrying the uh, another bacteria genes which will be transferred to the another bacteria. So bacterial genes can able to transfer from using the bacteriophage. It is happening in the nature, and we are utilizing this technique to transfer our gene of interest to our host or a bacteria. So here is the diagrammatic representation how this cycle is going on. A phage bacteriophage is there when it will inject its DNA. So that DNA will be a circular DNA, and this circular DNA can able to attach with the host DNA. So when the host DNA it will be attached, and when it will synthesize so many copies after the replication, and it will transfer its DNA within the folded in the bacteriophage, and new bacteriophage particle they are having the bacteria genome, and some genes which can be inside of the bacteriophage. So these new bacteriophage particle they are also having uh, your bacteria genome. and some portion only, not many, because the inside the bacteriophage very less space is there, so few genes only can able to transfer from one bacteria to another host cell. So when they will infect to another bacteria, so these bacterial genes, they can able to transform from one bacteria to another by the help of bacteriophage. This is the method where the bacteriophage is utilizing for the transferring of the genes, so from one bacteria to another bacteria. Now many bacteriophage is having their own host. See here, lambda bacteriophage is there, their host is E. coli. P22 type of bacteriophage is there, and their host is Salmonella typhimurium, and they are the specific type of host, and a specific bacteriophage is required specific host. They are not infecting with the all type of bacteria. Some specific bacteriophage, they are infecting specific bacterium only. Okay, then type of transduction. So two type of transduction is happening: specialized or restricted transduction and generalized transduction. So first we will see the specialized or restricted transduction. In this type of transduction, the phage transfer only few restricted genes. Not every genes they are transferring. Some specific genes only they are transferring. So if the bacteria is having these restricted type of genes, then only they will transfer, otherwise they will not able to transfer. Okay, so in this type of transduction, specialized type of some few genes only can able to transfer from one organism to another organism, where the generalized transduction, any type of gene can able to transfer from one organism to another organism. So first we will see the specialized type of transduction or restricted type of transduction. So restricted type of transduction only required some specific gene to transfer it. Then only they will be effective. Otherwise, if those genes is not present, the specialized transduction or restricted transduction will not be happen. So example is lambda and 580 type of phage is there. They are doing transduction through the lysogenic phases, and they required very specific sites. If specific sites is there, then only they will bind inside the bacterial genome. Otherwise, they will not able to bind it. So these specific type of genes, if it is there in the bacterial DNA, then only they can able to bind. So these specific type of DNA, when they will able to bind in between the DNA, then when they are creating their own copies, so they will take those genes which is there in the genome with the bacteria, newly creating bacteriophage, and when after the lysis of the plasma membrane of the bacteria, these newly synthesized bacteriophage can able to transfer those two genes where they are having infection to the other bacteria. So new bacteria can have those type of new genes, which is the part of the old degraded bacteria. Okay, so the dead bacteria which have those two type of genes, they can able to transfer by using this new particular bacteriophage. Then specialized restricted transfer example we will see. See, first example is lambda phage. So lambda phage is a host is E. coli K12, and lambda phage is double stranded DNA. Basically, if it is inside the host cell, double stranded DNA. When it is in the cytoplasm of the host cell, it will be circular. So how it is happening? This phage particle is having circular, non-circular type of double-stranded type of DNA, and it is having cohesive end. See these ends of the DNA, double-stranded DNA is cohesive end. Why we are calling cohesive end? So end of these one linear DNA, it is open, it is single stranded, and in between these single stranded DNA you have double stranded DNA. So these single stranded DNA, they can bind with each other because they are complementary. So when the double single stranded DNA is complementary, they can able to rejoin and they will make the circular DNA. So cohesive ends can, if they are complementary, so they can able to bind with each other and it will make a circular DNA like a plasmid. Okay, so lambda phage, they are having single double stranded linear DNA in the phage, and when it will infect to the bacteria, it will be circular double stranded DNA. So 12 palindromic sequence will be there in the cohesive bands. So these sequence like GGG, CGG, CGA, CGT sequence will be there in here in the cohesive bands, and opposite sequence of this, it will be there in the in opposite cohesive band. So this palindromic sequence can able to bind with the each other, so it will turn into the circular double-stranded DNA. Okay, so this lambda phage DNA can able to cause infection only E. coli K12 type. If you are having E. coli K12 type, then only it will infect. So other type of bacteria which is non-competent type of bacteria. So a bacteriophage required a competent type of bacteria only. So it will not infect with other type of bacteria. So bacteriophage is penetrating in the E. coli K12 and it is infecting. When it will infect the uh, linear DNA, it will turn into the circular DNA, and it will integrate in the host chromosome by crossing over. So the host DNA and the bacteriophage DNA, lambda-phage DNA, it will go using crossing over to integrate it, and site of integration is very specific, and we know that biotin synthesis gene and galactose utilization gene is there, and these two genes, in between these two genes, this lambda phage genome will be integrated. So bacteria which is having galactose utilization gene and biotin synthesis gene is there, in between this lambda phage gene genome will be integrated, and when this bacteriophage or phage DNA will be replicate, so two thing will be happen. When this bacteriophage replicate, so that the half of the genes means gal gene will go into the one bacteriophage, or biogene will go into the another bacteriophage, because the gal gene or biogene can't able to go into the bacteriophage. The reason, because the size of the bacteriophage inside the packing material is very less, and so that only one time they can take one gene only. Either they have to take the gal gene, or either they have to take the biotin gene. So biotin gene they can take, or they can take the gal gene. So by the name itself, we can say that if they are carrying the galactose utilization gene in the bacteriophage DNA, then we are calling lambda D gal type of phage. If they are carrying biotin gene, then we are designated as lambda D bio gene. So it depends what type of gene they are carrying, so we will call them. See, bio D gene is there, or lambda D gal type of bacteriophage is there. So lambda gal type, they are carrying the gal type of gene, and the biotype of variety, they are carrying the biotype of gene, biotin type of gene. So when they will infect a new bacteria, this gene which is having biogene, so this biogene will be transferred to bacteria, and the gal gene can able to also transfer into the bacteria. So this process is happening, and this, when they are carrying the part of the bacteria, and they are really releasing some bacteriophage DNA inside the bacteria, this type of mechanism we are calling defective phages, because entire bacteriophage genome is not the part of the bacteriophage. Inside, some of the bacterial genes they are carrying, and remaining bacteriophage DNA, it will be remain inside the bacteria only. So defective phages we are calling. So when they will release from the bacteria, they can carry either the galactose gene or biotin. Any one gene only they can carry. Okay. Then next specialized type of transduction is happening in the 580 phase type of phages, and it is having host or competent cell of E. coli. So this is the structure of the 580 genome. It is this double-stranded circular DNA when it is in fact in the E. coli. When it is in the patch particle, it is not circular. It is a linear DNA, double-stranded linear DNA, and this specialized transduction we are calling because it will attach in between the lactose gene and tryptophan gene, in between the lactose utilization gene and tryptophan utilization gene. So here the host genome is having TRP gene and lactose utilization gene. So in between these two genes, these Phe80 genome will be integrated, and with the host genome it will replicate again and again. It will increase its copy number, and when they will release from the E. coli, so they will carry either lac type of gene or TRP type of gene. They will not carry these two genes, only one type of gene they are carrying. If the newly synthesized bacteriophage, if they are carrying the lactose utilization gene, then we are calling lambda D lactose. If they are carrying the TRP type of gene, then we are calling lambda D TRP type of Phe80 phage. So it depends what type of gene they are carrying from the one organism to another organism. They have to carry only one type of gene at a time. Either lactose utilization gene they can take, or tryptophan utilization gene they can take. So on the basis of what type of gene they are carrying, we are giving their name. Now next is generalized type of transaction. This type of transaction where the phage transfer any type of level. They are not the specifically attaching to a specific position. In previous example, we saw the bacteriophage genome is attaching only in the specific location. There are two gene they will cut in between, and in between those two gene only it will. But in the generalized type of transaction, the bacteriophage DNA, it will attach in between the bacterial DNA and any non-specific type of manner. They don't. It's not specific. So generalized transduction, when it is happen, the bacteriophage DNA in any position in the bacteria. Okay. So example of P1 and P22 type of phage is there. They will attach anywhere in the bacterial DNA, and they can do for Lytic cycle and the Lysogenic cycle, and they will be a template phase only. So when they will infect in the bacteria, the viral genome may or may not integrate in the host cell. If it is integrating, then it will go for the uh, lysogenic cycle. If it is not integrating, and it will replicate, and they will synthesize new type of bacteriophage, and they can carry gene from one organism to another organism, then Lytic cycle will be there. Okay, so it means each and every genes has equal chance if they are not specifically transferring their bacteriophage DNA into the bacterial DNA. So every gene can able to have chance to go from one organism to another. Tell you, bacteriophage P1 type is there, which is having E. coli host, and E. coli host only specific, and not other bacteria. They are having infection of the P1 type of phage, and they are having double stranded type of DNA. And when double stranded type of DNA is there in the P1, they are non-circular type, like a thread type of structure. And when they are infecting in E. coli, they will be circularized. Okay, and they will not integrate into the host genome, but they will multiply by separately. So because they are not integrating in the specific site of the host genome, so that they will multiply as much as many times. Okay, so when they will penetrate of the DNA in the host, it degrade host genome into the fragments, and these phage replicate and also transcribe to produce mRNA for the capsid and protein production. So capsid and protein, other type of protein they require to produce the structural type of protein, and these structural type of protein can able to be formed the structure of the bacteriophage, because only DNA is not required. They also required some uh, structural type of protein to assemble the phage DNA into the phage particle. Okay, such phage when infect another host, then it transfer the gene from previous host, and these carrying genes they can able to transform from one organism to another organism. So generalized transducing phage we are calling when they are following this generalized type of mechanism, where they are not specifically integrate into the bacteria. Now, frequency, if you will see, the generalized transaction is very high, 10 to power 6 to 10 to power cells. They are doing this type of generalized type of transaction. So many organisms they are having infection, bacterial organisms they are having infection of generalized type of transaction. Here in this diagram, they are showing how generalized type of transaction is happening. See, gene A and gene B is there. These genes can be any one gene. It is not a specific type of gene. It may be any type of gene where the bacteria is having, and when bacteriophage is infecting, they can able to take any type of gene in the bacteriophage, not a specific type of genes they can carry. And when these genes is transferring is in the packed with the bacteriophage, when they will infect new type of bacteria, these genes can able to transfer into the new bacteria where these those specific type or another type of genes will be there. So a mutated genes, or maybe this a specific type, a type of bacteria, a type of gene is not there. So this gene can able to transfer into another organism. Since these genes is not specific, that's why we are calling generalized type of transduction. Okay. Then another example is P22 type of phage, and host is Salmonella typhimurium. So P22 type of phage is infecting Salmonella typhimurium, and genome is double-stranded DNA. Same thing, double-stranded DNA packed into the P22 type of phage particle, and it will integrate its genome into the host genome, but integration site is not fixed. So that's why it is a generalized type of transduction. So they can carry any type of gene which is non-specific, and they will infect a new type of bacteria, and they can transfer the any gene which is present in the into the host. The next type of transduction is abortive type of transduction. And this type of transduction, the phage genome has no capacity to integration into the host cell, and also lack genes for its own replication. So it remain as in the host cell. So abortive type of transaction means once they will integrate, they will transfer their genome into the bacteria, they will not integrate because they don't have capacity to integrate, and they don't have capacity to replicate their own. So what they are totally depend on the bacteria uh, enzymes, bacterial enzymes, and their mechanism. When the bacteria will replicate, their DNA also replicate, and they will transfer from organism to organism when they are transferring their own. Okay, so when bacteria is replicating, their DNA also will replicate separately, and when host cell divide, then it will be transferred to one and to the daughter cells. So mother cell, if this type of phage genome is there, so daughter cell automatically will receive this type of uh, phage DNA. So this type of transaction we are calling abortive type of transaction. Then next is phage conversion. So these phage conversion is change into the phenotype of infected bacteria and is referred as a phage conversion or lysogenic conversion. So lysogenic conversion or phage conversion is happening only as long as phage or prophage is present in the host cell. So when phage and prophage is present in the host cell, then only lysogenic conversion is happening. Otherwise, it will not happen. So some lysogenic phage may carry genes that can enhance the virulence of the bacterial host. And if those genes is not there, then the virulence of the bacterial host it will be affected. For example, some phage carry genes that encode toxins, and these genes, when integrated into the bacterial chromosome, can cause the once harmless bacteria to release some dangerous type of toxins. And these type of toxins genes can able to transfer by the this specific type of carrier phage. So the ability of the toxic genes which is transferred from new bacteria, they can also able to produce those toxic type of molecules, and they will be toxic type of bacteria. So when a bacteria is having toxic type of genes, and it will be transferred to the transduction or a specific type of phage, so the new bacteria which is having the new type of uh, characteristic, they will convert into the toxic type of bacteria. Okay, now lysogenic conversion. For example, we will see some bacteria. Here is the one example. Vibrio cholera is there, and it will be attacked by the CTX phage type of particle, and cholerae toxins, they will transform from one organism to another through this CTX phage. See, other bacterium E. coli is there, and lambda phage, it will be infected, and these Shigella-like toxins can able to transform from one organism to another organism. So many example is there where the a bacteria genes is transferring from one organism to another organism. Okay, now some important questions we will see. So, through transduction, when a recipient bacterial cell pick up bacterial DNA, what we are seeing. So a option is the new DNA replicate every time the recipient undergoes multiplication. B option, the DNA is destroyed. C option, the DNA modified to match and complement that of the recipient. The new DNA should enter a new bacteriophage, or it is lost. So one correct answer you have to choose. Then second question, a specialized transducing phage that attack an infected cell with the lysogenic stage of the same virus produce. Then A option is HFT we are calling, or B option HFR, C option F, or D option F minus we are calling. So any correct answer you have to choose. Then next third question. This is the route undertaken by the bacteriophage in transduction. What route will they are taking? A episome, B is recipient, C is donor, and D is vector. So correct answer you have to choose. The stability of cells to uptake DNA fragment from surrounding is known as HFR. A option. B option is the competence, and C option is facility. Then the option is fitness. So any one answer you have to choose it. Then next question, who discovered transduction? So Arnosky, Head and later Brand and Woolman, Griffith and later and Zinter. Then this process involved transferring naked DNA fragment between bacteria. A option is vectoring, B option is transformation, C option is transduction, D option is conjugation. The next question, this is the virulence of viruses producing bacterial cell after infection. If a specialized transducing virus attack a healthy bacterial cell, then what will happen? So four options is there. A option is virulence decrease there. Then B is equal virulence seen as the original one. Then C option more virulence than the original one observed. Then D option no virulence will be there. The next question, it is the when the phage introduces those bacterial genes only which are adjoining to the bacteriophage in the bacterial chromosome. So what type of mechanism it is? It is a conjugation, restricted type of transaction, or a specialized type of transaction, and generalized type of transaction. So what type of transaction it is? Then next question is, this is a incorrect regarding the specialized type of transaction. So you have to choose a one option which is not regarding the specialized type of transaction. Then next question, the viruses is used for transaction is lambda phage, T7 phage, T4 phage, and T2 phage. You have to choose the correct answer.